Hello student, today we discuss about the types of immunity that comes under the topic serology and immunology. So in this unit we mainly discuss about the what are the types of immunity and which organ, molecules and cells help in providing the immunity. So now we start. First, we have to understand what is the immune system and what is immunology, what is immunity and how the immunity responds. So, first this all term you have to understand. So, immune system is nothing but it is a cell, tissue and molecules that mediate resistance to infection. The cells, tissue or molecules which react when some foreign antigen or molecules interact with our body and our body react towards that foreign molecules, that is known as immune system. Clear? That is known as immune system. And immunology is the term which includes the study of structure and function of that immune system. What? Which include the study of structure and function of the immune system. That comes under the immunology. And immunity is when our body provide resistance against that foreign particles. When our body provide resistance against foreign particles, molecules or pathogen or anything like foreign particles, that resistance is known as immunity. And immune response, when our body react toward the foreign particles, molecules or pathogen, that reaction is known as immune response. Clear? That reaction is known as immune response. Collective and coordinated response to the introduction of foreign substance in an individual mediated by cells and molecules of the immune system. In simple words, in simple language, the reaction or response towards the foreign particles, the response of our body toward or against the foreign particles, pathogen, that response is known as immune response. Clear? So, what is the role of immune system? The main role of the immune system is defense against microbes, defense against the growth of tumor cells, kill the growth of tumor cells, right? What is the response? What is the response towards the foreign particles? How we react towards the foreign particle and how we defense against that microbes? How the body provide immunity against that foreign particle? These all are the role of the immune system to react or kill that foreign molecules, kill that foreign pathogen, so that they can't interact with our body, can't harm our body. Immune system act as defense system. Like our country, the border of our country is protected by army, right? The army is act as a immune system, because they defense our country against terrorists, right? According to like that only, immune system defends our body against micro pathogens, tumor cells, like that. These are the role of the immune system. Homeostasis means destruction of abnormal or dead cells. Dead cell or white blood cell, antigen antibody complex. They form antigen antibody complex and remove that dead cell. Clear? This all are the role of immune system. There are three types of molecules, organs or cells that comes under the immune system. That's organ, cells and molecules. The first organ that comes under the immune system that react against the foreign particles is tonsil and adenoids. Tonsil and adenoids. Thymus cells, most important, lymph nodes, spleen, Peyer's patches, appendix, lymphatic vessels, bone marrow. These all are the organs that comes under immune system. And cells are lymphocytes, monocytes, macrophages, granulocytes. In lymphocytes, T cell, B cell, plasma cell, natural killer cells, this all are comes under lymphocytes. And monocytes and macrophages. Granulocyte cells में in granulocyte cells neutrophil, eosinophils, basophils, this comes under granulocyte cells. Clear? And the molecules that act as defense against the pathogen are the antibody that's secreted by the B cells. Clear? Complement system, cytokines, interleukin and interferon. These all are the molecules that defends immune system, our body against the pathogen, react with the foreign molecules and kill them or form a complex and remove from our body. There are two types of immunity: innate immunity and acquired immunity. Clear? Innate immunity, that is non-adaptive immunity, means the immunity that present in our body from the birth or the first line of immune system is known as innate immunity. These are non-adaptive. First line of this is innate immunity is known as first line of immune response. Remember this. Innate immunity is known as first line of immune response. And this relies on mechanisms that exist before infection, that exist in our body before infection. This are not adaptive immunity, means when infection will take place and after that this immunity will be occur. That immunity is known as acquired immunity, and this is the second line of immunity. If this innate immunity is fail, then this acquired immunity will be response. This is the second line of response, and this relies on mechanisms that adapt after infection. And this acquired immunity is handled by T and B lymphocytes. T and B lymphocytes that produce antibody. What? These B lymphocytes produce antibody, so that they act against that foreign particles, pathogen, to kill them or remove them from the body. And acquired immunity is also a one cell determined, one antigen determinant, means one cell, one antibody only determine one type of antigen, not more than one type of antigen. These are the key term of the acquired immunity. So now in detail we study the innate immunity. Based on genetic makeup, relies on already formed components, rapid response within minutes of infection. When infection will take place, within a minute the innate immunity will respond. This is not a specific immunity. Same molecule, same cells responds to a range of pathogen, means wide range of pathogen are responded by a same cell or molecules. So it is not a specific that one molecules will respond against one antigen or one pathogen. No, not at all. It has no memory. Innate immunity having no memory, means if once pathogen will enter in our body and they are killed by a innate immunity, then there is no memory cell will develop in our body. so that in second times of infection by same pathogen, the body will react and not that infection will occur in our body. No, not like that. They not having a memory cells and does not lead to clonal expansion. The mechanism of innate immunity. The main mechanical barrier and surface secretion of innate immunity is skin, acidic pH in stomach, and cilia. These are the main mechanical barrier of the innate immunity. Humoral mechanism are lysozyme that is an enzyme, basic protein, complement, interferons. These are the humoral mechanism, how the body or how the body responds in innate immunity against the pathogen. Cellular defense mechanism is natural killer cells, neutrophils, macrophages, mast cells, basophil, eosinophil. See this diagram, this is a natural killer cells, this is a eosinophils, this is the neutrophils and the basophil and mast cells, monocytes, macrophages. Clear this? What is the mechanism? What are the different types of barrier are present in innate immunity? First, in mechanical barrier. In mechanical barrier, skin, acidic pH, cilia. In humoral mechanism, lysozyme, basic protein, complement, interferon. And in cellular defense mechanism, natural killer cells, macrophages, mast cells, basophil cells. Okay. Now, adaptive immunity. Adaptive immunity is the second line of response. When innate immunity will not respond against the pathogen, then after that this acquired immunity will be develop their response, and this is called as a second line of response based upon resistance acquired during life. Upon resistance acquired during life means this type of immunity is developed after the birth or during the lifespan. Relies on genetic events and cellular growth. Responds more slowly over few days. Innate immunity responds within a minute, but this acquired adaptive immunity responds more slowly over a few days, and it is very specific, means one antigen or one molecules will respond only particular type of antigen, not a wide range of antigen. So it is a very specific, has anamnesic memory, repeated exposure leads to faster. First response is lower, after that they develop memory, and when second times that infection will take place, then they respond faster. Repeated exposure leads to faster, stronger response and leads to clonal expansion. These are the types of adaptive immunity. There are two types of adaptive immunity: active immunity and passive immunity. Active immunity having a clinical, subclinical and infection, and passive immunity is provided by a breast milk, placenta, right? And artificial immunity are provided in adaptive immunity by the vaccination. The types of vaccine is live, killed, purified antigen vaccine. And in passive immunity, artificial immunity is provided by immune serum, immune cells. These are the types. Adaptive immunity mechanism. What are the mechanism? How they react and how they form an antibody against the foreign particles. So, first is cell mediated immune response, that is CMIR, cell mediated immune response, and this cell mediated immune response is the response that takes place by the T lymphocytes. What? T lymphocyte eliminate intracellular microbes that survive within phagocytes or other infected cells. Clear? Cell, remember this, cell mediated immune response is response by a T lymphocytes, and this T lymphocytes eliminate intracellular microbes. What? They eliminate intracellular microbes that survive within phagocytes or other infected cells. Second is humoral immune response, and this response is mediated by B lymphocytes. Clear? There are two types of lymphocytes, B and T. T produce response against cell mediated immune response, and B is a humoral immune response and is mediated by antibody, because when differentiation of B cells which takes this, they differentiated into two types, plasma cells and memory cells. And that plasma cells, see here, this is a plasma cells, and these plasma cells produce antibody. Clear? These plasma cells produce antibody. This humoral immune response are two types. Not humoral. When B lymphocyte will differentiated, they are of two types, plasma cells and memory cells. These memory cells they remember that infection later, and this plasma cell produce antibodies so that they kill that pathogen. Clear? And so that's why this is mediated by antibody and eliminate extracellular microbes and their toxin. They eliminate extracellular microbes and their toxin. This is a cell mediated immune response. What? Cell mediated immune response. This is a T cell, because cell mediated immune response are mediated by T cells. Clear? This is, so what happened? They recognize peptide antigen on macrophage in association with major histocompatibility complex class. See, this is a antigen. Antigen are in the form of peptide, right? And these antigen are recognized by this class MHC. What? MHC. This is a major histocompatibility complex. They recognize this antigen on macrophages. Clear? And the processing will be takes place here. Antigen is processed. Identified molecules on cell surface helps body distinguish cell from non-cells. The main function of this MSC is to distinguish this antigen from self from non-self. Clear? What is the main function of this MSC? This is a major histocompatibility complex. They distinguish the molecules from self and non-self. If it is non-self, then the processing will be takes place. Antigen is processed. The process antigen and class first MHC. This is the process antigen and class first MHC. Cytotoxic T cells. T cell are of two types, TC cell and TH cell. TC cell is the cytotoxic T cell, this, and TH cell is the helper cells. So this TC cells receptor recognize process antigen. This process antigen are recognized by this TC cell, right? This is a TC cell, recognize this TC cells, and after that the process antigen recognize the process plus class first MSC. Clear? Then what happened? They activate helper T cells. Helper TH cell will be activated. Okay? Cytotoxic T cell become activated. Activated cytotoxic T cell process antigen and class first MSC, so that they produce molecules. They produce molecules like cytokines, this type of molecule, so that this antigen, the process antigen or viral protein will be killed off, and this viral protein will be die. These are the process. Clear? First, macrophages having a class first MHC, they recognize antigen peptide. This is self or non-self. If they are non-self, the processing will be takes place. They break down the polypeptides of the antigens. The processed antigen are recognized by TC cells. This TC cell recognized the process antigen and then they produce a cytotoxic molecules, right? Cytotoxic molecule are produced and this cytotoxic molecule kill that antigen. Clear this all process? So these are a process how the foreign antigen are killed. T cell goes into effector cells stage, that is able to kill if infected cells. Effector cell stage, and that is able to kill infected cells. Second, there are two types of T cells, T helper cells and T cytotoxic cells. So helper T lymphocytes that recognize CD4 T cells activate phagocytes to kill microbes, and cytolytic T cells, that is CD8 plus T cells, destroy infected cells containing microbes or microbial proteins. What? CD8 plus T cell destroy infected cells containing microbes or microbial proteins. Clear this? So cell mediated immune response are primary response, production of a specific clone of effector T cells, and memory cell, production a specific clone of effector T cell developed in several days. This response developed in several days does not limit the infection. Secondary response more pronounced, faster, because why secondary response are faster? Because during the primary response memory cells will be developed, right? Memory cells will be developed, so that during the second response the response will be faster, because memory cells will develop and they recognize that pathogen easily. More effective at limiting the infection. Example, cytotoxic reaction against intracellular parasites, delay hypersensitivity and allograft rejection. Now second is humoral immune response. What happened in humoral immune response? It is a B lymphocyte. What? It's the B lymphocytes recognize a specific antigen. Humoral immune response are a B cell types immune response. Remember in earlier slides I told you this, right? So B lymphocyte recognize a specific antigen, right? Proliferate and differentiated into antibody secreting plasma cell. Antibody secreting plasma cell. Antibody binds to a specific antigen on microbes, destroy microbes by a specific mechanism. Some B lymphocytes evolve into the resting stage and memory cells. See the diagram. This is the antigen, right? This is the antigen. This antigen this antigen having a peptides, right? This is a B cells. This B cell having class 2 MHC. This class 2 MHC recognize this antigen, this is self or non-self. If it is a non-self cells, then they process this antigen, right? During the processing of the antigen, this class 2 MHC break them into a peptide bonds in a small fragments, right? Antigen is processed. During the processing of the antigen class 2 MHC, processed antigen are displayed. The helper T cell activated. What happened here? Helper T cell will activated, and when helper T cell will activated, this B cell differentiated into plasma cells and memory cells. And these plasma cells produce antibody. What? They produce antibody, so that these viral particles or antigen will killed out, and the memory cells that help in recognizing this infection for later time. Clear? Now antibody. The antibody that produced from the B cells, what are the type? Belongs to the gamma globulin fraction. Antibody are also known as immunoglobulins. What? Immunoglobulins. It is antibody or Y-shaped structure or T-shaped polypeptides. It is a Y-shaped or T-shaped polypeptide. This is they having two identical heavy chain and two identical light chain. They having two identical heavy chain. This is a heavy chain and this is a light chain, and this is the Fab region where antigen are binding, and this is known as parato. Clear? And this is known as parato. All immunoglobulin are not antibody. All immunoglobulin are not antibody. There are 5 kinds of antibody, that is IgG, IgM, IgA, IgD and IgE. Clear? There are 5 types of antibody. So first is IgG. Actually IgG is having 70 to 75% of total immunoglobulin. What? 70 to 75% of total immunoglobulin and secreted in high quantities in secondary exposure. And this IgG cross the placenta. What? They cross the placenta. This is the only antibody that are present in a fetus, because this IgG cross the placenta. This IgG having a properties to cross the placenta and provide immunity to the fetus. Major function and application are they neutralize microbes and toxin, opsonize antigen for phagocytosis, activate the complement, protect the newborn. What? They protect because how they protect the newborn? The question will be asked, how the antibody IgG protect the newborn, because this is the only types of antibody which cross the placenta and they protect the newborn or fetus. Clear? The fourfold rise or fall indicates active infection. A single positive sample indicates past exposure. Clear? IgM secreted initially during primary infection. When infection will be takes place, then initially the antibody that are produced from our body are IgM, and after IgM IgG will be produced. Clear? That's why IgM is the initial antibody that secreted from the body, or it is the primary infection. Question will be asked, which antibody are produced during primary infection? IgM, IgG, IgD, IgE. Then right answer is IgM. This IgM not cross the placenta, because only the IgG, only the IgG that cross the placenta. The major function of the IgM is they secrete first during primary exposure and they activates the complement system used as a marker of recent infection. During the antibody test, they mainly test 2 type of antibody, IgM and IgG. According to this IgM concentration of IgM and IgG, they detect how long exposure will be takes place to that particular antigen. Next is IgA. IgA is monomeric in serum. IgM is a pentavalent. What? They are pentavalent. Presence in newborn means infection. Single positive sample in serum or CSF indicates recent or active infection, used to detect early phase of infection. IgA is a monomeric in serum, dimeric with secretory component in the lumen of the gastrointestinal tract and in the respiratory tract. Major function neutralize microbes and toxin. This is a monomeric in serum. This is the main function of IgA is in the gastrointestinal tract and in respiratory tract. And serodiagnosis of tuberculosis. During the tuberculosis, they detect the IgA. Synthetic respiratory viral test. IgD. IgD is the monomeric, it is also a monomeric. Only IgM is the pentameric, right? IgD is the monomeric. Major function and application is present on the surface of B lymphocytes, function as membrane receptor. Main role of IgD is not clear. Has role in antigen stimulated lymphocyte differentiation, but main role was still not clear. Ig is the major antibody. It also mediate types. First, hypersensitivity. They respond against the allergy. When allergy will be takes this, then the concentration of IgE will be high at that time, and this is also present in monomeric form, and major application associated with anaphylaxis, and they play a role in immunity to helminth parasites. Sequential IgM and IgG humoral response. IgM produced as a first response to many antigen. Already I told you IgM is the first response, and IgG is the produced after IgM, or is called as second response. The level of IgM high transiently. IgG produced after IgM, and higher level persists in a small amounts throughout life, produced in large amount during secondary response. During secondary response, the concentration of IgG will be high. What concentration? See this graph. This is the graph. This is the first time of infection, and this is the second time of infection. During the first time of infection, the concentration of IgM will be high, right? High. When the concentration of IgM will be low, after that the concentration of IgG will be high. But during the second response, when second time reaction infection will takes this, the concentration of IgG will be high. What concentration of IgG will be high? There is no increase in level of IgM. So first line of defense is IgM, and then after IgG. Clear? That's IgG produced in large amount during secondary response. Persistence of antigen sensitivity memory cells after primary response. Failure of immune response. Immune response helps individual defend against microbes, some cancer cells, right? Immune response can fail in hypersensitivity reaction and during immunodeficiency deficiency. In these two types, our immune system will be fail. Hypersensitivity reaction. Hypersensitivity will be act when allergy will be takes place. When our body reacts overreaction to an infection, that is known as hypersensitivity. Cause cells damage through excessive immune response to antigen, hypersensitivity overreaction to infectious agents, allergy overreaction to environmental substance, autoimmunity overreaction to cells, and immunodeficiency where our immune response will be fail, loss or inadequate function of various components of the immune system, and can occur in any part or state of the immune system, right? The immunocompromised host has an impaired function of immune system, is at high risk of infection. Congenital, it is a primary immunodeficiency, and acquired, it is secondary immunodeficiency. Like AIDS, chronic leukemia, it is a secondary immunodeficiency. Clear? So altered immunity, immunocompromised, this is the chart. Altered anatomic barrier, immune system, innate immunity, reduction of complement, activate phagocytosis, adaptive immunity. So you see this table, and remember how the innate immunity, adaptive immunity will be work, and which cell will be acts, and what is the function. So in this ppt what we study, we study about immunity, innate immunity, right? We study about innate immunity. It is the first line of defense, has no memory and non-specific mechanism. Second, adaptive immunity, developed entry of microbes into the host cells, and it has a memory to deal with subsequent exposure and happen to a specific cells, T cells, B cells, and having a memory cells. Third, primary immune response, that is by IgM, secondary immune response, that is by IgG, and failure of immune response can result in hypersensitivity and immunodeficiency. So these all are about the types of immunity that comes under the topic serology and immunology. So today we stop here. Hope this topic are clear to you all. Thank you.